Hello, everyone. Welcome to Wiley and welcome to our webinar. My name is Tom Machinchik and I'll be your host. We have a great presentation for you today. The title is Cryo-correlative electron tomography for structural biology of complex mixtures. Our speakers today will be Nicholas Ariotti. He's group leader at the University of Queensland, and Dr. Ling Bo Yu. She's a senior product marketing manager, materials and structural analysis division at Thermo Fisher Scientific. Now, before I turn it over to our speakers, I'd just like to remind everybody that you can ask any questions that you might have for the speakers today. If you see the chat box on your screen, just type the question that you have into the chat box at any moment during the webinar, and we will answer your questions at the live Q&A when the presentations are over. Also like to remind everybody that this event will be available on demand after the presentations are done. We will send you an email where you could follow the link and watch it again, or you could send it to one of your friends to watch it for the first time. Now, without further ado, I'd like to turn it over to our speakers and let's get started. And I'll see you back at the live Q&A when the presentations are over.
Well, thank you for the kind introduction. I just want to start by saying thank you to Wiley and to Thermo Fisher Scientific for making this happen today. It's great to be able to highlight some of the work that we've been doing recently in my lab at the University of Queensland. So in very general terms, the immune system is broken down into two subsystems, the innate immune response and the adaptive immune response. Innate immunity involves germline-encoded pattern recognition receptors that can respond very rapidly to challenges by PAMPs and DAMPs, but this response is general and works only against classes of pathogens like viruses, bacteria, or fungi, but not specific pathogens. The adaptive immune response takes much longer, but it is highly specific to antigens and epitopes that recognize signaling molecules and molecular signatures on individual pathogens. For today's talk, I'm going to focus specifically on the innate immune response, namely the NLRP3 inflammasome. My talk today is targeting one specific pathway of response, and that is the NLRP3 inflammasome response. When cells are challenged with a particular PAMP or DAMP, it primes a transcriptional response that results in upregulation of a protein called NLRP3, as well as cytokines like IL-1B, IL-18, and interferon beta. After priming, there is a second step, termed the activation step, which involves a series of diverse signals converging on the activation of the protein NLRP3. When NLRP3 is activated, it undergoes a conformational change from an inactive donut-like structure to an active pinwheel-like structure that recruits other proteins, including apoptosis-associated speck-like protein containing a card, or ASC, and procaspase-1 to form a massive intracellular complex. You can see ASC up here in red. These are the specks that it forms in the cell, and here are the core protein constituents of this intracellular macromolecular assembly. Ultimately, the stimulation and activation of NLRP3 results in the activation of the pore-forming protein called gasdermin D, which is cleaved and activated and inserts into the plasma membrane, where it oligomerizes and causes the rupture of the cell in an inflammatory process called pyroptotic cell death. And you can see here, up in green, we have a cell that's undergoing pyroptotic cell death. And just like that, the cell has died. We now have a detailed structural understanding of the differences between the inactive and active NLRP3 complexes. Here we have two examples of this structure, with the inactive double ring cage coming together with interdigitation of the leucine-rich repeats with inactive domains at the top and the bottom of this cage. Just 2 years ago, this paper was published in Nature from Zhao et al., which really elegantly solved the organization of the active form of the NLRP3 complex. Critically, the structure shows that you have the directed arrangement of the pyrin domain on one side of this particular complex, which is free to interact with other pyrin domains, just like on the ASC protein. So what do we know about ASC? Well, ASC is a 22 kilodalton protein made of 2 domains linked by a disordered region. The N-terminal pyrin domain is comprised of 6 alpha helices. We also know from structural studies that I'll get onto in the next slide that the protein forms these complex interaction interfaces that interact to generate a filamentous-like arrangement. Mutations that disrupt this interaction interface impede the protein's ability to form specks in cells and, in some cases, filaments in vitro. And in the interest of time, I won't get into this today. There's also the caspase recruitment domain, which is comprised of 6 helices also. We also know that mutations in this domain can affect normal spec formation in cells, which together indicates that both domains, the N and the C, the pyrin and the card, have a role in higher order ASC multimerization. Finally, there is the disordered linker that spans between these 2 domains. This disordered linker is highly flexible. We know that the removal of this domain inhibits spec formation in cells, but not filament formation. This potentially indicates a role in the branching of this particular protein complex. Okay, so what we know about the higher order structure of ASC filament comes from the cryo-EM of the pyrin domain. Initially characterized by Ed Egerman's lab over 10 years ago now, they showed that the pyrin domain has C3 symmetry. It forms a helical core assembly with a central pore of around 20 angstroms and an outer diameter to this pyrin domain filament of around 90 angstroms. This was validated just a year later by Saborghi et al. But critically, the structure of the full-length ASC protein, that is the N-terminal pyrin domain, the linker, and the C-terminal card, hasn't been able to be solved in vitro. More recently, Jorgo Modis's group at the LMB demonstrated that they could resolve ASC filaments within the cytoplasm of bone marrow-derived macrophages from mice in situ, but they couldn't generate a high-resolution reconstruction of these filaments. All of this comes together to say, so far we have no full-length structure of ASC in its filamentous forms. Groups have tried recombinant expression of this complex, and they haven't had any success. Similarly, we've had no success so far from in situ analyses. So are there different systems that we can use to get at this structure? Well, recombinant expression is the normal approach that people start with when it comes to solving the structure of a protein of interest. This is the system that was employed to solve the structure of the pyrin domain, and it's a very traditional recombinant system where you transform your plasmid, you induce the expression of that plasmid in your host E. coli, you lyse your cells, and then you perform significant purification steps. And this can generate large amounts of protein at high purity, but it's really important to note that this isn't the only approach that is available. In the past, we've had success with this kind of method. This system exploits a eukaryotic parasite of lizards called Leishmania tarentulae. And with this system, we can culture the promastigote form of the Leishmania. We can lyse these cells. Then we can add plasmid directly to this lysate with some cofactors. And within 3 hours, we can have very high quality protein produced. This system's been used extensively for characterization of protein-protein interactions and protein dynamics using single molecule detection methods like fluorescence correlation spectroscopy and fluorescence cross-correlation spectroscopy. Its major advantages are that it's fast to generate the lysate and it can be prepared quite simply. It's a eukaryotic cell, so it's great for eukaryotic protein folding. And as I said, it's been used for stoichiometry analyses. You can use it to look at protein dynamics, and you can modify proteins with post-translational modifications. And these are all things that we have done in the past with this system. But like with any method, there are in fact caveats. Here we are dealing with very small volumes. This is 20 to 50 microliters of protein at a time. We have a lower protein yield and we have low protein purity. So very low volumes of protein in a complex mixture is quite a problem for traditional structural approaches. So what do I mean by that? Well, this is what a traditional structural approach looks like. It's much easier to do these kinds of analyses on highly homogeneous preparations of proteins. And as such, cell-free expression and its application to structural biology have so far been reasonably limited. It's important to note, however, that alongside the developments in single particle methods in cryo-EM, in the last 10 years, there has been the advent and application of the entire field of in situ structural biology. And the tomogram that you're looking at is the first ever in situ tomogram that I acquired, and I think it nicely highlights the difficulty of working with these kinds of samples. There is just so much information, there is so much crowding of the structures, it's really highly concentrated, and it's really hard to discern the individual events within the volume that are your structures of interest. Identifying your structures of interest is incredibly challenging. So how can we do this? Well, there have been methods that have been developed. And this comes to the crux of what we are trying to achieve with our work. Can we use in situ cryo-electron tomography-based targeting approaches to broaden the applicability of cell-free expression systems to structural biology? This is something we published in the Journal of Cell Science last year, where we expressed a fluorescently labeled protein that localizes to microtubules, and we can target those individual microtubules in the cell using cryo-electron microscopy. So we now have a method to target our structures of interest. Can we get in there and start doing structural biology with cryo-CLEM to specifically target our macromolecular complexes from our cell-free expression system? In order to do that, we need to have a better structural and ultrastructural understanding of our starting material. So we started by looking at the Leishmania tarentulae cells and then looking at the lysate. Now, Leishmania tarentulae are apicomplexan parasites of lizards. They have quite a complex life cycle. They are transmitted from geckos and other lizards to sandflies through a blood meal, where they transition from an amastogote aflagellated form to a promastogote flagellated form, and this is the form that we culture. You can see here in this red box. They're simple but very interesting cells. They have abundant endoplasmic reticulum, Golgi, mitochondria, and strikingly abundant lipid droplets with a large flagellum. But I think the most distinctive feature is the subpellicular microtubules that are arranged all over the surface of these cells just below the plasma membrane. And you can see this arrangement here. What's incredible is that we can see a regular protein arrangement decorating these microtubules, even in conventional fixed transmission electron microscopy approaches. Okay, so we know what the cell's like. What do the actual lysates look like? Well, we can do some negative stain on this particular component here, and we can get a sense of what's present. Unsurprisingly, the lysates aren't very clean. There's a diverse range of structures present. We have cytoskeletal elements, including actin filaments and microtubules. We can see abundant ribosomes in these negative stains, and strikingly, we can see a lot of vesicles as well. Now, this gives us a broad view, but we really wanted a clearer view of what was present in this lysate, so we undertook an unbiased cryo-electron tomography screen of this lysate. Just like you would for any protein preparation and single particle analysis, we took our lysates, we put them on the grid, we plunge froze them, and then we transferred those grids into our Glacios 2 cryo-electron microscope. This microscope's been fitted with a Falcon 4 detector under the control of the latest version of the Thermo Fisher software for tomography. And this is regularly what we generate in terms of data on the system. We can acquire dosimetric tilt series and get around 150 to 250 tilts per day. The most we've acquired in one sitting is a little over 450 tilts over a weekend. And then we have a data processing pipeline set up on our local HPC cluster. Okay, so we acquire our dosimetric tilt series, we tilt the stage at our position of interest, and from that, we generate a series of back projections that we can reconstruct later. We can use serial parallelization acquisition in the tomography software, which really increases our throughput here, we're really improving sort of 8 to 10-fold in terms of the number of tomograms that we can acquire relative to the old way of acquiring tomograms, which is 1 at a time. And then we can go through and we can batch reconstruct all our tilt series with IMOD or eTomo. And then finally from there, we go through a subtomogram averaging pipeline, which I'll talk about in a little bit. Okay, so we have our unbiased structural characterization of the Leishmania tarentulae lysates. But what does that actually look like? Well, we have large vesicles and we have small vesicles present in our lysate. We have incredibly abundant ribosomes. This is a good thing because we're expecting to use this as an expression system. We have actin filaments, abundant microtubules, as well as lipid droplets present. So we can actually look at whole organelles that are intact using this system. It's just a quick outcome of the kind of screen that we did. And what was quite striking was that the microtubules that were present were reasonably abundant. They were present in 20% of all of our tomograms, which was a little bit surprising to us. Okay, so this is a tomogram of an area here. What I hope you can see are these abundant microtubules present. This is quite interesting because these cells are cultured at 26 degrees. They're lysed at 4 degrees. We then snap freeze these samples, we thaw them, and then we store them on ice prior to plunge freezing. We haven't done any Taxol stabilization of these microtubules whatsoever. These microtubules from the Leishmania tarentulae are incredibly thermally stable, unlike mammalian microtubules, where if you fluctuate the temperature, they completely depolymerize. So we know we can lyse our cells, we know we can resolve microtubules. The next question is this method applicable to generating good quality reconstructions of our protein present in the lysate? Can we get a high resolution reconstruction out of this way of processing our cells? This is a very brief introduction into our data processing pipeline. I won't spend much time on this. If you want more detail, I'm happy to answer questions afterward or via email. Basically, we're using a workflow where our Glacios acquires our tilt series, and then from there, we can screen for quality based on automated batch reconstructions using eTomo. From there, we go through Motion Core implemented in Reliant 5, and then through an initial refinement at bin 4 in Dynamo with particle picking in Cryolo. From there, we can extract all those particles and work through our reduced bin data up to bin 1, and we can do a CTF refine and Bayesian polishing for our final post-processing. And this is what it looks like. This is the data that we generate on our Glacios 200 kV system. I have to stress that this is without an energy filter. So this is 29,000 particles acquired at 73,000 times magnification with a pixel size of just under 1 angstrom. What I hope you can see here in blue is the density of our reconstruction, our map, and on the panel on the right, we've got the blue and the orange. These are AlphaFold models of alpha and beta tubulin that we are able to dock into our protein density. So we used AlphaFold because this Leishmania tarentulae alpha and beta tubulin. All of this gave us a final resolution of 7.1 angstroms, which is what I think is quite impressive given how our setup is quite basic. And I think this nicely highlights the quality of Glacios that you can use very simply and easily. Okay, so we know we can get to reasonably high resolution with this data, but can we use the expression of fluorescently labeled proteins for expedited structural biology in our cell-free system? We know that these systems are low yield, they're expensive, and they're not biochemically pure, but does cryo-correlative fluorescence microscopy-based targeting overcome the issue of yield and purity? From this work, our collaborators show that they can generate large molecular weight structures of ASC in cell-free lysates. They believe that these ASC specs, that they call them, resembled ASC formation in the cellular context. So we wanted to test this, so we went through a negative staining protocol, and we showed that the pyrin domain, GFP alone, resulted in significant filament formation. I hope you can see that there. We also showed, as we zoom in, that these filaments look very normal relative to what other people have generated previously. But critically, when we express the full-length human ASC in this system, we form these very dense uranyl acetate rich structures that when we zoomed in, we could resolve extensive filaments protruding from the edge of these domains. So to check that these ASC filaments are in fact our full-length ASC protein with our GFP tag, we performed immunoelectron microscopy with an anti-GFP antibody directly conjugated to gold particles. And this is what that looked like. You can see that these filaments extend from a dense core, and at low magnification, this closely resembles what we see for ASC filaments, ASC specs, in the cellular context. When we zoom in, you can see that these ASC specs and these filaments are directly labeled with gold, but our microtubules up here in the top right are completely devoid of labels. So we really know that these are, in fact, GFP positive and are our ASC filaments. Okay, so this is the workflow that we've generated that we've termed CC-Flexit for correlative cell-free leishmania expression and cryo-electron tomography. Basically, we can take our crude lysate after 3 hours of expression and freeze it directly onto our cryo-EM grid. This allows us to then target our complexes of interest by cryofluorescence microscopy. Then we can move on directly onto cryo-electron tomography. It's important to note that all of this can be done within a single day. You can start your expression in the morning, then plunge freeze after 3 hours and do some fluorescence-based targeting around lunchtime for data acquisition on the cryo-TEM overnight. And this is what your data can look like. This is what our data looks like. This is the expression of a protein of interest. This one that targets to microtubules. It's a mutant of CAVN1. As we zoom in, you can see that each area, there's abundant fluorescence in all of these areas and all of these grid squares. And zooming right into one area, we can see that this corresponds with densely packed microtubules. Now, we're not 100% certain that this particular protein is associating with these microtubules. We're in the process of validating that and the structure of this particular protein at the moment. So this is what CC-Flexit of ASC full-length GFP looks like. We can see this is a cryofluorescence image of a grid imaged at liquid nitrogen temperatures on a Lincoln stage. What I hope you can see here are very bright specks of fluorescent ASC. We only have 300 odd specks in this entire grid, and only 70 of them are in areas that we can target with our cryo-electron microscope. So these specks are far too infrequent to look at with any other technique other than cryofluorescence microscopy and correlative microscopy. Also, they're far too infrequent to do any sort of traditional structural approach like single particle analysis on to get to high resolution. There's just not enough of them. It's important to note that these specs look very similar to what we've characterized previously by our traditional EM approaches and what others have seen in cells. So we can go through the workflow and start to characterize the structure of these. This is an atlas from our Glacios where we've overlaid our ASC fluorescence signal. We can zoom in on individual grid squares. We can highlight our areas of interest that we want to analyze by cryo-electron tomography. We can generate individual search maps, which allows us to be very accurate with where we're positioning our tilt series. And then from there, we can go through and look at these areas with high GFP fluorescence. And from our cryotomograms, after reconstructing these tilt series, I hope you can see that we have abundant filaments all through this particular area of ice, which corresponds to our GFP, our ASC full-length GFP. So we went ahead and applied our structural pipeline on these AF filaments. What we have here is a very interesting structure. To look top down at the ASC, it actually looks quite like a flower. It's unfortunate, but this didn't get to very high resolution, only around 20 angstroms. We think this has more to do with the inherent flexibility of the protein and its promiscuous behavior than our ability to acquire good quality data. So this is what it looks like when we apply no symmetry whatsoever. If we apply helical symmetry, however, we can see that this map starts to better resolve. And if we go through and we apply C3 and helical symmetry together, we can get a really well-defined map. What I hope you can see here are these ring domains around the outside of our ASC filaments. We think these are in fact the beta barrel of the GFP. So we think we can place the GFP, but unfortunately the resolution here is just too low to be very definitive about anything here. So this is the overall structure that we've resolved. We have a flower-like structure that is 210 angstroms wide. This includes an N-terminal pyridine domain, a linker domain, a C-terminal card, and a C-terminal GFP tag. Importantly, the central pore resembles exactly what others have characterized previously for the pyridine domain. We've got 20 angstrom spacing between our central helical arrangement of our pyridine domain. Okay. So not only can we generate a structure here, we can use this CC Flexit system to gain more insights into the biology of this particular protein. Others have shown previously that ASC can form intricate branching networks in situ. You can see here, this is a segmentation of these branch points in orange. Interestingly, we can also see that in our tomographic data. If you zoom in, you can have a look at these individual branch events that I've highlighted here with these black arrowheads. They're incredibly abundant in this data. Interestingly as well, this isn't the only bit of biology that this system can tell us about ASC. This same paper looked specifically at these ASC filaments, and they noted that in situ, ASC associates in its filamentous form with ribosomes. And you can see here we've got these hexagonal representations of where these ribosomal associations are. When we look specifically at our ASC filaments in our cryotomograms, we can also see abundant ribosomal association with our filaments. As you can see here, we've also segmented that, and there is just an incredible abundance of ribosomes associating with our ASC filament. We actually think this is part of the reason why we can't get to a higher resolution to our structure of interest. Okay, here's a few more examples of exactly that. You can see ribosomes directly associated with this filament. So we went on, we solved the structure of the Leishmania tarentulae ribosome from these data sets. We didn't go to a high resolution. All we wanted to know was, can we resolve the large subunit and the small subunit, and can this start to tell us a little bit about this interaction between the ribosome and the ASC filament? So we finally took a deep dive into our averages that are present in our tomograms, in our reconstructions. And what we noticed was that when we separate out these different averages, we had a few different classes that showed a potential association between the ribosome and our ASC filament. So we focus specifically on this ASC filament with this ribosomal association here in class 3, and we tried to push this down a more structural route. Unfortunately, it just wasn't possible. So we went through a more curated approach. We picked out 100 particles, and we looked at the structure of what we could resolve from those 100 average particles. And what we saw was quite interesting. This is preliminary data, but what you can see here in gray is our ASC filament, and in pink we have our ribosome. And as we apply and we tighten the mask on our ribosome, what we can start to see is that the large subunit is facing our ASC filament and the small subunit is facing away. So we think this gives us a potential interesting structure to pursue in the future, and we're currently working on this at the moment. So I just wanted to finish now with a very quick summary. Here we've developed a rapid method for proteins expressed in the Leishmania tarentulae system that allows for structural biology. We can use cryogenic correlative light and electron microscopy to target proteins expressed with fluorophores and then do subtopogram averaging on that. We've used the Glacios Thermo Fisher microscope to solve the structure of the Leishmania tarentulae microtubule to 7.1 angstroms. And finally, we've characterized ASC spec formation in the Leishmania tarentulae lysate. We formed the first structural full-length analysis of this ASC filament, and we noted a branching phenotype and an interesting association between ASC filaments and ribosomes. So with that, I just want to thank all the people involved. A massive thank you has to go to Dr. Vikas Tulu, who's been dealing with my questions about this project on an hourly basis for the last 18 months. A massive thank you to all the members of the Ariotti lab, the Parton lab at UQ, the Collins lab also at UQ, the Gambin lab at UNSW, and a big thank you to the good people at Thermo Fisher, especially Jessica, who helped us with the segmentation of the ASC filaments. And with that, I would look forward to receiving your questions.
Hello, everyone. Following Nick's fantastic results, I want to today briefly talk about a couple of design details we spent our efforts on to enable high quality tomograms on Glacios cryo-TEM on our microscope. So this is a beautiful example of a tomogram, which shows a lot of cellular context. For example, this Golgi apparatus. Although rich in cellular context, the field of view of a tomogram is actually small. For example, this tomogram has a field of view of 1.2 micron. 1.2 micron is only a tenth of a lamellar surface. And a lamella is only a very thin slice of a cell, and only a partial cell, not the whole cell. And a cell on a TEM grid is these tiny blobs. So the original field of view of a tomogram on this graph, on this slide, is almost invisible. It's really small, but how small is it? Let's imagine a TEM grid is now the Earth. Equivalent view of a tomogram on the Earth is roughly 4 kilometers. What is 4 kilometers? That's actually the length of the Central Park in the New York City. Now imagine taking all these pins, take one of the pins and put it exactly on the Central Park on this globe. That's really difficult, right? Well, one would argue your globe is too small. I need to zoom in to see. That's exactly what we do in cryo-EM to find the area we are interested in. Starting from the atlas of the grid of the TEM grid, equivalent to the view of the Earth. And then we zoom in to find the New York City, equivalent to find the cell on the grid. Then the New York City, equivalent to the view of a lamella. With that, we can finally put our pin on the Central Park. Therefore, in our TEM tomography acquisition software, we need to define 4 optic presets to build a map. The 4 presets corresponds to the grid atlas, and a preset to allocate a cell, a preset to define a detailed lamella map, and then the final optics to collect a tomogram. Now we have a tomo pyramid defined. The next question is, how often do we need to use the pyramid? Very frequently, it turns out. First, of course, we need to use a pyramid to locate the exact destination, like finding the Central Park, as in the previous example. This is a difficult task. Fluorescence labeling to help targeting the protein of interest, like Nick showed, can be helpful in this workflow. And second, when navigating to the destinations, we also need to use the pyramid view. For example, traveling from the Central Park to the Opera House in Sydney, we can't just go there. We need to zoom out first to locate Australia, then the city of Sydney, and then the Opera House. So this frequent optic change poses specific design challenges on electron microscope. What challenge is that? Now let's step back a little bit to see what's the basis of an electron microscope. So this is a classic ray diagram of a light microscope we all see in our high school textbook. Light gets bent when passing the lens and focus onto the focal point. The electron microscopes share the exact same principle. The only difference is we don't use lens. We use electromagnetic lens, which is a coil. When a current goes through the coil, it creates a magnetic field that can bend the electrons. So the magnetic field is nicely demonstrated in this picture on the right. We are putting a lot of current in order to bend the electrons with high energy, 200 keV or even 300 keV. High current generates high heat. Changing optics causes temperature fluctuation, then leads to instability in the column, such as mechanical drift of the specimen. In cryo-EM, we are very aware of the impact of a drift or motion. Demonstrated in this paper, image processing steps such as motion correction nowadays can of course compensate a lot, but still it is better to start without the unwanted drift after optics change. The way we solved the thermal drift is a very neat trick called the constant power lens. Our lens contains two sets of coil instead of one. The currents are running in the opposite directions, creating the magnetic fields also in opposite directions. The delta between these two fields is the effective field to the electron beam in the end. This way, we can control the lens to any desired strength while keeping the generated heat a constant. Constant power lens means no temperature fluctuation, means no drift, and instant high resolution images. Now we have crafted our lens to be able to do the frequent optic switch. We are able to find and go to any exact destinations. We should tilt our stage and collect the tomogram. But after stage tilting, we don't just collect the image. We have to do two more steps. That's tracking and focus. Why do we need to do tracking? In the right figure, we are showing a stage. We imagine it just tilts along the tilt axis. But in reality, when tilted, it also moves in X, Y, and Z. This will move your feature in the field of view. For example, the left image is, say, before tilting, and now after tilting, you can see the feature or the white cross has moved. Now let's see the effects in a complete tilt series. The middle is the raw tilt series. So you can see how jumpy it is. It is all over the place. And then the right part is a lined tilt series. At a certain point, our feature is completely lost. Then you can see you only see the gray images. Instead of collecting images at each tilt to fill the resolution in Z, we are simply wasting time here. So this particular tilt series will not result in high resolution tomograms. Therefore, tracking is critical in tomography data acquisition. We normally have a tracking image taken after tilt, but the tracking image is not infinitely large. If we're too far off, no overlap features can be found in the tracking image, we will lose track. Therefore, we have a critical specification on our microscope called the stage geocentricity. This defines the maximum stage displacement within the complete sweep of stage tilt range. And then here I want to emphasize that this is a good example of hardware and software working together as a system. We need to tweak our hardware, but hardware is never perfect. especially we're talking about the nanometer accuracy, we need the software to fix the remainder. But software can only fix a certain amount. Hardware still needs to be good to start with. And this slide looks almost identical to the previous. Beside X and Y, the stage also moves in Z. As a result, we need also an additional step to refocus. For example, we're showing a focus image here. And similarly, this focusing image is not infinitely large. It poses a requirement of how much we can allow Z to change. I know from Nick's previous experience, he had some quite difficulties to collect the proper tomograms due to the stage issues. The stage centricity specification is tuned and checked on our system, not only in the factory, but also upon installation, if tomography package is part of the configuration. So in my opinion, this is critical to have for high quality tomogram data acquisition. And in summary, tomography is like a real estate problem. It's all about location. Find exact location, go to the exact location, and check and refine the location after stage tilting. We have a tomo pyramid to facilitate these tasks. We have also introduced constant power lens on our microscope to allow frequent optics changes. We have stage centricity which is critical for feature tracking after stage tilting. And as a system, hardware and software are working together to fulfill the task. With that, I think I would like to say thanks for your attention and looking forward for the Q&A session.
Hello, everyone. Welcome back to the presentation. Thank you, Dr. Yu and Dr. Ariotti, for your amazing work in presenting it here at Wiley. I can't believe the scales that you guys operate on. It's just tremendous. Anyway, we do have a bunch of questions from the audience, and I'd like to get started with the first one. This one is for Dr. Ariotti. Have you modeled your ASK complex using AlphaFold, and what did you see?
Yeah, that's a great question. We have tried. I would say that AlphaFold did an okay job, but it's a little bit confusing as to what it generated. So I wouldn't say I'm an expert in AlphaFold, but I know that there's a kind of a limit to the amount of amino acids that can be modeled in that system. We've got our ASK, which is 22 kilodaltons long, about 200 amino acids. And I think as a maximum, we can have about 25 ASK full-length protomers modeled in AlphaFold. When we do it for lower molecular weight kind of complexes, we model sort of up to an 18-mer. What we have is kind of an interesting arrangement of ASK that doesn't really make much sense. So we'd have sort of a planar array of our pyrin domains associated with one another, and then on the opposite side we'd have our cards associated with one another. It's not until we get above an 18-mer that we start to generate sort of a filament-like arrangement that we observe with our cryo-tomograms and our reconstructions. So I think a 21-mer, 22-mer, and 24-mer all form this quite nice filaments with the pyrin domain arranged as the core of the protein and the card domain decorating around the outside. From memory, the 22-mer was certainly the thing that modeled closest to what we generated in our reconstructions and our model. But unfortunately, the resolution of what we achieved from our reconstructions with ASK wasn't really good enough to accurately fit any of that modeled AlphaFold model into our density map.
Okay, thank you, Dr. Ariotti. You mentioned that you collect 400 to 500 tomograms per weekend. What factors do you believe contribute to such high throughput?
Yeah, so there is one major factor here, and that is the software itself. So the tomography software has implemented serial parallelization for its acquisition of tilt series now. So it used to be that you would take a single tilt series in a single position and then move to the next spot and take another tilt series. The way that tomography acquires the data, it's kind of inspired, I think, by Radonev's PACE Tomo, is that we'll have a series of areas that we can set up as acquisitions, and we take a tilt image and then we acquire more areas, and then we tilt and we acquire more areas. And this way it really allows us to improve our throughput sort of 8 to 10 fold. So where we would normally take one tilt series, now we take 10 in the same amount of time. So that's the major advantage. Another advantage is kind of the way that we acquire our data. So we're always doing correlative microscopy. So we're doing fluorescence-based targeting of our structures of interest, whether that be in situ or, in this case, in our cell-free lysates. And by having fluorescence targeting, we can really only image the areas that we know are going to have our target in it. So we don't just do needle in a haystack. We don't randomly image. We're imaging our areas that we want to image. We know that our targets are going to be present. So that improves our acquisition time in the microscope, no wasted acquisitions, but it also improves our downstream data processing, where we're not wasting our time looking for things that aren't present in our data.
Okay, great. This one is for Dr. Yu, this question. Is it correct that it is only the objective lens of the glaciose column that is constant power? Changing the magnification has large effect also on the condenser and projection lenses.
Yes, you're absolutely right. So on glacials, we only have constant power lens for the objective lens, while on cryos we have constant power lens for everything. And you're also right that when changing the magnification, it also affects the condenser lens and the projection lens. However, the effect is much less, and that's why we chose to only use a constant power lens for the objective. Because the major image distortion, aberrations, for example, coma, astigmatism, they all come from the objective lens. And then the other two lenses, because sample is not stored in those planes, so it doesn't also introduce the sample drift, which we observe, which in our observation is the biggest problem from the heat that generates. On the other, for the condenser and then for the projection lens, it has other issues. That's true, and that's our challenge to solve next.
Okay. Does the glaciose have another stage than the Titan Krios? Does that question make sense?
I guess you picked out that I have two rows that we have a different number for cryos and glacials. So the technology is the same. It's both are called compute stage. So we use the same technology. However, the models are different. So we have a different Mark 1. We have a Mark 1, and then cryos has a Mark 2 compute stage, and that results in the spec difference from for the eccentricity, which listed on my slide.
Okay. This is for Dr. Ariotti. What other equipment do you have except for the Glacios 2? For example, cryofluorescence microscope, high pressure freezer, etc.
Yeah. So for the lab, yeah, we have high pressure freezers, and we also have plunge freezers. So we have a Leica EM GP2, and we have a Vitrobot as well. So for this workflow, we have a Zeiss LSM 900 Eriscan 2 upright confocal microscope. So this allows us to do sort of 2 times resolution, 2 times improved resolution imaging compared to standard confocal. Now this microscope is fitted with a Lincoln CMS 196 stage, which we use pretty much for all of our core correlative analyses. Very nice microscopes. The advantage of the Lincoln stage, I think, is that we can do 3 grids at a time, and there's quite nice integration with the Zeiss software so that we can really automate our fluorescence imaging as well. So we basically load our grids, we set it up to acquire those 3 grids, and we can leave it for 2 hours and then come back and it will have imaged everything that we wanted to image. So in terms of what we have for this workflow, I would say it's reasonably basic. So plunge freezer, cryofluorescence microscope into a cryo-TEM. We're not using fib or anything like that.
Okay. Doctor, you mentioned that you faced difficulties in your previous experience. Could you elaborate on the specific challenges you encountered?
Oh, I think that might be for me, that one. So Lingbo alluded to some of my problems in her talk. So for a good period of time, we tried very hard to acquire tilt series and tomograms on Joel cryo-EM, and while it does perform reasonably well for single particle analysis for tomography, it just really wasn't up to the task of what we wanted to do. So for subtomogram averaging, structural analyses, we need to be able to really do batch acquisition. The quality of the stage of what's present on the cryo-EM, maybe I would say, is not reliable enough to do batch acquisition. So if we want to acquire at our specific region of interest, with that microscope, we found that we just couldn't walk away. We had to sit there and watch everything because it would tend to move and not accurately re-register the position that we started, that we set to acquire. So it really wasn't feasible getting any sort of really, I guess, high throughput analyses on that microscope with the way that the stage behaves.
Okay. Could you comment on the complexity of sub-tomogram workflow? The packages you mentioned are new to me. How challenging is it to learn them?
This is a question that I receive every time I kind of present this data. First off, all the software that we use is in the public domain. So these are all freely available that you can download and get working basically today. I would say it's challenging to do these things, but certainly not impossible. And this is something that myself and my postdoc, Vikas Tolu, have been working on for the last couple of years. So we start with kind of batch reconstruction. So we acquire our data on the microscope. We batch reconstruct in eTomo or iMod. And this then is really just about quality controlling our data. So we can reconstruct, we can screen those tomograms, which ones reconstruct well. Lingbo went through this in her talk. Which ones don't, we discard those that don't. Then we go through kind of a second set of quality control, which is, do these contain our proteins of interest or do not? Discard those that aren't relevant. Then from there we import we go back to using our tilt series, and we'll import them in through Reliant 5, where we'll do motion core in Reliant 5. From there we'll take those tilt series and put them into Dynamo. And from there, Dynamo is really great for sort of low resolution initial refinements, and we'll do that in Dynamo. And we'll also use Cryolo. So Cryolo is a freeware software that was developed out of Stefan Raumster's lab, which really is quite fantastic at doing automated particle picking. You can train your own models. You can pick whatever you want. We use Cryolo to pick out our ribosomes and associated with our ASC filaments, so we can get good classes out of that And then from there, we go through the full Reliant workflow. We do this all in Reliant simply because something like Dynamo, it just doesn't have Bayesian polishing or 3D CTF refinement. So we can't really get to high resolution using that sort of software. And Reliant has some really nice tutorials that are available online, so you can teach yourself how to do these things. The tutorials are based around viral particles, but it's with a little bit of troubleshooting and practice and hard work and dedication, you can get it working.
Okay. One more question for Dr. Ariotti and then one for Dr. Yu. You mentioned that the data is collected without an energy filter. Do you anticipate achieving better results if an energy filter is used?
Yeah, that's right. So our system does not have an energy filter. We would very much like it to have an energy filter. The baby steps, we'll get there eventually. So a selectress, if we had it on our microscope, would certainly improve the signal to noise of what we have in our data, which would improve our particle picking. So ultimately, resolution is protein dependent. But if we had, say, a selectress set up and we could directly compare, say, our microtubule analyses between what we've done already and what we would have on a system with a selectress, we'd be very interested to see what sort of resolution improvements we could get simply by having that energy filter present.
Okay, and we only have a few minutes, 4 minutes left. This last question is for Dr. Yu. MAPS software is fantastic for correlative workflows, but is limited in its application to the tomography. With the significant investment that Thermo Fisher is making in the correlation space, i.e. integrated fluorescence and cryo-FIB, what is the next in the pipeline for Thermo Fisher tomography software? Is there any appetite for integrating correlative workflows into tomography?
First, I'm glad that you find MAPS fantastic. Of course, MAPS and tomography software are connected. MAPS sessions can be imported into tomography sessions. So I showed in one of the tiny images where you can have the fluorescence represented overlaid on top of the search map, which allows you to target your region of interest more accurately. And then one thing that we recently did on the tomography, specifically on the tomography side, which I'm also excited about, is we made a combined software package, tomography software. It combines all the existing elements that we had into one package. Not only it includes the data acquisition part, which Nick also showed with a very high throughput nowadays, and then also does a real-time tomography reconstruction. And then the third one is we also now extended the tomography data acquisition from one single grid to multi-grid, which means that we have a cassette, so you can basically set up the whole 12 grids and then leave for your holidays and let the microscope collect for a week to get you thousands of tomograms in a week. And then one thing we also introduced is something called the discovery view. So I showed this pyramid view. So discovery view allows you to browse your data afterwards with this pyramid view. And then about the future development about correlative, this is our new attempt or new initiative. We now only do the discovery view with the TEM data, of course, but we want to bring in all the other correlative aspects into the database so we can better view how this data are overlaid on top of each other. But of course, we just started. We're we're still in the early phase. If you have a specific request, feel free to reach out to us, and that will also help us to shape our development for the future.
Okay, great. Well, we're almost out of time here. Have less than a minute left. I just want to say thanks again to Dr. Yu and Dr. Ariotti. Super interesting presentation today. I didn't understand half of it, but I don't work in the field really. But anyway, I was amazed at the scale that you guys work on. It's just super amazing work. Anyway, so if people in the audience had a question that we didn't get to, we will get to you through email and send an answer to your question. Please don't forget that this video will be available on demand if you want to watch it again, or if you want to share it with one of your friends. And so I'm going to end here and just say thanks once again to Dr. Yu and Dr. Ariotti, and hope to see you on the next webinar series. Thank you again for attending, and we'll see you next time.
Thank you.
Bye.